Okay, good morning from New Orleans. I'm Jason Gardner, and I'm glad to be speaking to you today about my research. And so the focus of my laboratory work is changes in the extracellular matrix in the heart and how that it plays a role in the pathogenesis of heart failure. So I'll start with a bit of career progression. So that slide come up. Okay, so I was actually born in Baton Rouge, and about fourth grade I moved to Mississippi and went to public school there and through high school. So really neither Louisiana or Mississippi really claims me as a native. Okay, so but I got back to Louisiana when I went to school at LSU in Baton Rouge. And I have a bachelor's in chemical engineering, and I was all set to work in a chemical plant. I actually had quite a few job offers, and I even did a year rotation at Dow Chemical there in Plaquemine. But I wanted to further my education, and also was very interested in biological processes, which I hadn't been exposed to very much in my undergraduate career. So I chose to go to graduate school in biomedical engineering at Louisiana Tech University, which is the same university where my LBRN student Keshab is from. And so I graduated with my degree in engineering and decided to take really more of an academic research career path, which really requires some postdoctoral training, which is basically like a really intensive research program. You get paid, but you don't get paid very much. Okay, it's really low pay, but you learn a lot about different research techniques. So I did a postdoc fellowship at the University of South Alabama School of Medicine, and that's in Mobile, and learned about smooth muscle reactivity and studied really microvessels. So there was a lot of microsurgery that I did in that lab. So my first faculty position was as a research assistant professor at Auburn University at the vet school. I don't know if you know where Auburn University is. I mean, it's in Alabama, but I've got a lot of people that have trouble picking it out on a map. Okay, it's almost to the Georgia border. It's not too far from Atlanta, actually. So there I studied, I worked with Dr. Joseph Janicki, and he is internationally known for his studies in cardiac mast cells and heart failure. He really got me interested in this whole extracellular matrix remodeling of the heart, and taught me a lot of the different in vivo techniques that I still use today in my own research. So I then worked for a few years at the University of South Carolina at the School of Medicine there in Columbia, and in 2008 I took my current position as assistant professor at LSU here in New Orleans. So we'll shift gears and talk about my research a bit. And I'm trying to stay away from too much data, so I want to give you some basic information. All right, Keshab advised that I not show too much data. Okay, even though that's really what I'd like to show. But let's start off with what is heart failure. Okay, so heart failure doesn't mean a heart attack. It doesn't mean that the heart has stopped beating. It's just the inability of the heart to pump enough blood to really satisfy the metabolic needs of the body. So it's just not moving enough blood. And when it doesn't move enough blood, the blood tends to pool up in different parts of the body. So you've heard about pulmonary edema. You've seen people that sometimes have a lot of edema in their extremities, like in the feet and such, and that can be caused by heart failure. So what's the cause of heart failure? It's really any type of damage or overworking of the heart. So that can be caused by a heart attack, which we call a myocardial infarction, where a big portion of the heart has actually died and it's no longer functional. So the heart can't perform appropriately. It can be due to sustained hypertension. So if you have hypertension, it means that the heart has to work really hard to move the blood. This causes the heart to change its structure. It grows, it gets a lot more collagen in there. And we'll talk a bit more about that in the next few slides. It can also be some type of a birth defect, valve disease, coronary artery disease, and even actually diabetes is becoming a big problem as far as heart failure is concerned. So it's a progressive disease where the mechanisms that kind of keep your heart functioning properly actually exacerbate the condition and actually make the disease worse. So just some statistics. This is, I believe this is 2004 numbers, so these are probably worse now. So each year there's about almost 600,000 new diagnoses of heart failure, and these are U.S. numbers. There's a really very, very large prevalence of heart failure. This means like how many people are actually living with this condition, and it's about 5 million right now. And the annual mortality is roughly 300,000, and it's actually increasing. Now, it's largely a disease of the elderly, so about 10% of people over 65 years have heart failure. And there are some gender differences. I mean, men have a higher mortality rate than women. But women tend to live longer than men in general, so there's a very large female population of heart failure patients. So that human cost also comes with a dollar figure as well. I mean, this is very, very expensive condition to treat, and it tends to be a chronic condition. So once people start treatment, it goes on for 5, 10, 15 years. So this is an annual cost of about $38 billion, and it's roughly 5% of the healthcare budget. So this is a huge, huge clinical problem. So when I talk about extracellular matrix remodeling, or remodeling in general, so what are we talking about? Well, it's cellular and structural changes that the heart undergoes in response to an insult or injury. So if there's some type of damage or overworking, say like hypertension, if the heart's having to work too hard to push blood out of the heart into the blood vessels because there's excessive blood pressure, then there's changes, or this remodeling occurs in the heart. So the cellular changes. Now, you've probably heard of hypertrophy, right? I mean, it's an increase in muscle mass. So it's just like if you start lifting weights and you want to get all buff for the beach or whatever, and you increase the size of your biceps. Well, if you work your heart, it will increase in size the same way, but it's not necessarily a good thing. I mean, there's an exercise hypertrophy, which is pretty good, but in this case, this is a pathologic condition. So pathologic hypertrophy is never good. Dilation, in here it is not a typographical error. This dilatation is the process of dilation. So that's an increase in the chamber volume. That just means the inside of the heart gets bigger so it can hold more blood. So there's cellular alterations involved in this, and there's also extracellular alterations. And the extracellular matrix alterations that's what I really study. So all of these changes that are occurring in the heart, these are part of a compensatory mechanism, meaning it's going to allow the heart to push more blood and maintain what we call cardiac output, or blood delivery to the body, over a period of time. But that initial compensation actually becomes a problem, and it becomes pathologic over an extended period of time. So all these processes are actually good processes that you need, but they become bad if they're chronically activated. So what the heck is the extracellular matrix? Let me let that slide come up. So here is, so you can see that, can't really see my arrow on the screen, but anyway, so what you're seeing here from left to right is a picture of a rat heart that is being digested, that the cells are being removed using a detergent. And as the cells are removed, you see this white, all the way on the right, you see kind of this white, opaque looking heart, and that's the extracellular matrix. So the heart is primarily composed of, the extracellular matrix anyway, is primarily composed of collagen 1 and 3. Those are the fibrillar collagens, we call them, and those are the ones that provide like structural integrity for the heart. Also provides structural integrity for a lot of different organs, but we're talking about the heart today, okay? The ECM also, there's a bunch of different types of other things, collagen, fibronectin, laminin, elastin, but the big players are the collagen 1 and 3, and that's really what we focused on in the lab. Now, to study these extracellular matrix changes in heart failure, we use a couple different animal models. And so what I'm going to talk to you today about is a rat model of volume overload. And so this is a surgical model. And so what you see on the screen is actually the abdominal aorta, which is on the right. It's that reddish large artery with a needle going into it, and the vena cava, which is on the left. So we create a shunt or a fistula between the abdominal aorta and vena cava using a large needle. So what this does is causes a shunting of blood. So shunting of blood from the aorta into the vena cava. So about 30% of the aortic blood will go right back into the vena cava, right back to the heart, and we call this a volume overload. So it stresses the heart. And this will cause a lot of remodeling, some cellular remodeling, some structural remodeling, and eventually this will cause heart failure in these animals. And a lot of the things we see as far as growth factors and cytokines that are induced in hypertrophy is very similar to what's found in human heart failure. So here I couldn't resist, I got to show you some data. Here's some examples of the cardiac remodeling post-fistula. Let that come up. So first off, I'm showing a graph of cardiac output. So you have the sham, which is your control animal, and the fist, which is the volume overload animal. And this is just showing that cardiac output is significantly increased due to that shunting of blood. And the little probe I'm showing you there is how we measure that. That's a Doppler flow probe. So that little hook you see that's kind of metallic looking to the left of the blue stuff, that's actually hooked around the aorta, and it uses the Doppler method to estimate the flow of blood through the aorta, and that's an index of cardiac output. So it's very increased in the volume overload model. Now here's some, on the right it's a dime and a sham heart and a fistula or volume overloaded heart. And you can see just the gross change that's occurred. The volume overloaded heart is huge, right, compared to the sham, because it's been overworked, so there's some hypertrophy involved. There's also some dilation of the ventricle. And this is a graph of the LV mass that gives you an idea of how much the LV weight, the left ventricular weight, has changed. And so it's much greater in the volume overloaded or fistula animals. And here's a picture of some perfusion fixed hearts and that come up. On the left, we have a sham animal, which is your control, and on the right, we have a volume overload animal. And you can see the dramatic differences in the size of the left ventricular chamber, which is on the right in this picture, and also the right ventricular chamber, which is on the left, kind of bows out on the side there. I don't really have a way to use a pointer over here to show you exactly what I'm talking about. But anyway, you can see there's a lot of gross anatomical changes that have occurred due to the volume overload. Now, one of the techniques we use, this will be a challenge. Let's see, this is videos. One of the techniques we use to track function and changes in the ventricular chamber is ultrasound echo. So it's the same technique as is used in humans clinically, but we do it on rats. Now this is not looking too pretty on y'all's end out there, but what we see on the left is a sham, and it's much smaller than what's shown on the right, which is the volume overloaded animal. So the videos aren't looking too good on that end, but anyway, this is one of the techniques we use to measure cardiac function non-invasively and also changes in chamber size. It's very nice that it's non-invasive because we can track these animals over time, which is very useful in our studies. So instead of hitting you with a bunch of data, I'm going to talk about some of the techniques we use in the lab. I'm going to have some interesting animations, but they're probably not going to work too well. So we have, like I said, we use surgical models of disease in rats and mice. I've only talked about the rat model today, but we also do similar types of techniques used in mice. And one of the models we use in mice is actually a pressure overload surgery where we constrict the aorta. So it just models different types of human disease, where the volume overload is more like what you would have in some type of valve insufficiency, like mitral valve prolapse or something like that. The pressure overload model is more akin to like aortic valve stenosis. So there's several different animal models we use in the lab to study the progression of heart failure. We do a few different in vivo assessments of cardiac function. We talked about the echo. That's the picture you see on the right. We also do, that's non-invasive. We also do some invasive measurements. We talked about how we measure cardiac output using that little Doppler flow probe. And we also do some catheterizations of the rat heart. The picture of the catheter, that's what's held between the thumb right there that little picture on the left, that's a catheter that we feed down into the heart by way of the carotid, like through the neck of the animal. And there's pressure volume loops, that we call it, and that's those functional measurements you see in the blue, and you've got the smaller, the green sham loop, and you got the red volume overload or fistula loop, and it just shows that that animal has a much larger heart and also has some depressed function as well. So these are different techniques we use. We also do a lot of different in vitro assays in the lab. On the right, we have an isolated heart preparation that we can use to perfuse different types of agents into the heart directly to avoid any type of systemic changes in the drugs. So this focuses on just the heart itself. In the middle and to the left, those are cardiac fibroblasts. So my lab studies extracellular matrix. Well, the fibroblasts are what make the extracellular matrix. So we do a lot of studies, a lot of culture studies looking at fibroblasts and trying to figure out different pathways involved in the regulation of collagen. And of course, we do a lot of molecular and histological assays. We do Western blotting. We do histology of stains of collagen. That's what you see in the red there. That's actually a picoserious red staining of a blood vessel that's got some fibrosis around it. And of course, we use on the right, it looks kind of like a Western blot, but that's actually a zymography that's measuring MMP activity. And that little thing that says sham, fist, prolox, and lox, that's a Western blot. So unfortunately, we have to do a lot of Western blots, and it kind of drives the students crazy, but it's a good measure of protein changes in the heart. So I just want to tell you a few things about one of the current projects, and this is a project that Hisham is involved in the lab. Is the role of lysyl oxidase in the progression of heart failure. So LOX, lysyl oxidase, is a collagen cross-linking enzyme. Without LOX, the collagen stays soluble and it never becomes deposited into the tissue. So LOX is a necessary thing, but it looks like in heart failure that it becomes overactivated and overexpressed, and it leads to fibrosis and other bad things. So in a fairly recent clinical study, they found increased LOX expression in failing human hearts. And so we kind of ran with this and wanted to look at LOX in our disease model. So in this graph, this is echo data. The blue is a sham. The red is the volume overload of the fistula, and this is time on the x-axis. On the y-axis, it's an indicator of the LV chamber size. You see that the volume overload causes a progressive increase in chamber size over time. And this is not a good thing for the heart. When it gets too big, it functions poorly. And here is a measure, in the same animals, a measure of function, of systolic function, by way of echo. And you see that the systolic function is depressed in those volume overloaded animals. Now, if we measure lysyl oxidase expression by way of Western blotting in the hearts from those animals, we see that the sham animals is not really much different, so they're all the time points are pooled. But we see a progressive increase in the lysyl oxidase expression in those volume overloaded animals, with really the highest expression occurring in the failing hearts, which is that 21 week or that last time point. And even though I'm not showing you the data, one very interesting thing we found is if we use a LOX inhibitor, we can prevent this progression of dysfunction in these animals with the LOX inhibitor. And what's probably even more interesting, if we allow the animals to develop some cardiac dysfunction, we can actually reverse some of that dysfunction. Okay? So you might be thinking, well, why don't we go ahead and let's use this in humans? Well, the problem is the drug that we're using, or the agent that we're using to inhibit the lysyl oxidase, is actually inhibiting lysyl oxidase all over the body. So it's a systemic inhibition. And LOX is very, very important for collagen deposition. So LOX is very important in bone formation and bone function and just maintenance of bone. So you can't just throw this inhibitor onto humans and expect good things to happen. So we need to find a way to target the lysyl oxidase specifically in the heart, and we're just not there yet. So to learn more about the mechanisms of how this lysyl oxidase might actually improve what we call ECM metabolism or collagen metabolism, we study cardiac fibroblasts. Now, cardiac fibroblasts, they're the key regulators of the extracellular matrix. They produce the extracellular matrix, collagens and such. They produce growth factors that influence the production of extracellular matrix, and they also produce lysyl oxidase and TIMPs, all that go towards ECM synthesis. So the ECM metabolism is always a balance between synthesis and degradation. So the fibroblasts can also produce MMPs, which degrade collagen. So here's the basic concept of what we're doing. Let's let that pop up. So that the stress, some type of stress in the heart, we're just calling it like a physical stimuli, actually causes the fibroblasts to produce growth factors, cytokines, even extracellular matrix like collagen and lysyl oxidase as well. So these growth factors and cytokines that are produced actually have an autocrine effect, which means they affect the fibroblasts. So if the fibroblasts produce a growth factor, they can respond to that growth factor. So you can think about there's some positive feedback loops that can actually become uncoupled here in disease states. Those growth factors and cytokines also affect the cardiomyocytes, so we would call that a paracrine interaction. Some of these things are good and some of them are bad. Anything in excess is typically bad. And so using the cardiac fibroblast culture and some co-culture techniques that we're developing in the lab, we hope to learn more about how this whole regulation of extracellular matrix is working in the heart. And so here's one last bit of data I'll show you. These are cardiac fibroblasts we've isolated from control animals and volume overloaded animals and age-matched shams. And we see that the volume overloaded fibroblasts from the volume overloaded heart actually have a much greater expression of lysyl oxidase. And so this is an important aspect of our study, and we're actually going to use cells from the diseased animals to learn more about how the extracellular matrix is regulated. So just a quick acknowledgement slide. We've got a lot of people that helped develop all the data, a lot of people that have worked with me over the years, but Jessica and Milo, they do a lot of the animal model work and surgeries with me. Tatiana did, she's a postdoc, she's pictured on the right, left-hand side of that right panel. She did the Lox activity assay and a lot of the culture. And then we've got a couple of summer people in the lab, Kishab in the top left, and a few other folks as well. And I have to acknowledge the funding source is right now I'm funded by the American Heart Association and also on by the NCRR through the COBRA program. So I'll be glad to take any questions you may have.
Do you have any questions in your audience there? Let's start with your own audience.
Does it look like it? Oh wait, we have one. We have one.
So far, what are possible ways to prevent bullying?
So the question is, what are some possible ways to prevent volume overload? And so right now, really most of the clinical treatments for heart failure involve reducing the workload on the heart. And one of the key ways to do that is to reduce blood volume. And so almost everybody that's got any, I'd say everybody, let's just say everybody that's got heart failure is going to be on a diuretic. That's one easy way, cheap way to reduce blood volume and reduce the workload on the heart. But like with a lot of treatments, clinical treatments for heart failure, it's just management of symptoms. And so what we're hoping to do is with the extracellular matrix studies is to find ways to really target the initial cause of heart failure and try to reverse those changes so that the heart can function better.
Just like the kidney transplants, they also like could you transfer some parts of the heart, like from someone who is like disease, like transplant a good part of the heart from a good person to someone who's infected?
So yeah, the question is about cardiac transplant. Yes, and so they do cardiac transplants, but it's really, there's only so many organs out there, and not everybody's going to be a good candidate for a heart transplant. I mean, it's a very, you can imagine, I mean, it's a very rough procedure. There's a lot of anti-rejection drugs that have to be taken, and there's just not a lot of hearts out there that are going to be available. So, hopefully, eventually, hopefully, we can actually engineer new hearts. I mean, we're a long way from doing that, but that's a good thought. Maybe one day.
All right. So, is that why it costs so much money to work on the heart? Is that why it costs so much to treat a heart failure patient?
Oh, so one reason why there's such high costs of treating heart failure patients is that there's so many of them. I mean, there's millions and millions of heart failure patients, and you're talking about it's really a condition more than a disease. It's caused by many different types of diseases, and so patients will live with heart failure for many, many years. I mean, you can live for 20, 30, maybe even more years with a heart failure diagnosis. I mean, some people are diagnosed very young with heart failure, and they don't know why, what damaged the heart, like idiopathic, what they call it, cardiomyopathy. And if you get diagnosed with heart failure, the drugs can actually make your heart function normally, but you have to stay on those drugs for really the rest of your life. So that's one reason why it costs so much. Oh, we got one more over here.
Go ahead.
Tell me a little bit more about the reverse dysfunction of lysyl oxidase. How does that work?
So yeah, so Esau was asking about how the lysyl oxidase reverse the dysfunction, and I have a couple theories about that. So we know that there was some extracellular matrix changes that occurred that turned the diseased animals, turned their hearts back into really more like normal animals as far as the extracellular matrix composition. So we're talking about collagen, really. Okay. Now on top of that, we think there's functional changes in the myocyte that are occurring as well, and I believe that when you inhibit the lysyl oxidase, you actually reduce the oxidant load on the heart. So the whole oxidative stress level is reduced in the heart. And oxidative stress is really damaging to the myocytes. So I think that's another reason, and we're actually looking into that right now. We really don't know. That's brand new data. So I think that's all the questions on our side.
Okay. How about Shreveport? Oh, we can see you. No? Children's Hospital, you two there? Have any questions? At least shake your head, yes or no. Okay. Over here now. No questions. All right then. Young?
Well, thank you, Dr. Gardner.
Thank you.